Thanks again for the kind introduction. And as Lou already told you, I'm today explaining you a bit about CRISPR-Cas and its impact on today's world with the title A Revolution in Genetic Engineering. So in recent years, CRISPR-Cas very much dominates the scientific media landscape with bringing up headlines like CRISPR-Cas9, the gene editing tool that has revolutionized molecular biology, starting controversies like the CRISPR baby scandal, what's next for human gene editing, but also giving glimmers of hope, like CRISPR takes its first steps in editing genes to fight cancer. And these are only some of the headlines that can be collected in order to explain how groundbreaking CRISPR actually is. Not to forget, of course, the Nobel Prize in Chemistry that was awarded to Emmanuelle Charpentier and Doudna in October 2020. And my aim today is kind of to bring you CRISPR-Cas a bit closer, tell you what its function is, and also explain to you why it's such a groundbreaking biotechnology. And in order to do that, I want to talk about a genetic disease that where a lot of progress was made in the last few months. And this is about hemoglobin disorders. So normally, red blood cells are the vehicles of oxygen within our bodies, having hemoglobin as the molecule that binds the oxygen and releases it at the organs that require the oxygen, actually. So you can think about hemoglobin in a way like a mailman who is picking up the packages at the reception point, like the lungs, and delivering it or driving in the post vehicle, the red blood cells, and delivering it to the organs, or delivering it to the customer, who then are the organs receiving the oxygen. Unfortunately, there are some persons who have a mutation in the gene encoding for the beta subunit of this hemoglobin, the gene called beta globin. And this mutation causes the abolishing of the production of beta hemoglobin, therefore leading to a defective form of hemoglobin that only contains the alpha subunits of its molecular structure. And this reduces the efficiency of oxygen binding, and it also leads to a strong deformation of the red blood cells. And you can imagine, staying with the example from previously, you can imagine it like the mailman, for example, is catching the coronavirus and can't work anymore. So someone else has to be exchanged for someone else who is now taking over the job, which is unfortunately someone from the office who doesn't know how to drive the van, and he also doesn't know the fastest way to go to the customer. And this is actually taking place with the hemoglobin. The oxygen binding is reduced, and from time to time, there are crashes that lead to severe symptoms. And these severe symptoms include organ dysfunctions like jaundice or cirrhosis, but they can also lead to a strong expansion of the bone marrow that eventually leads in the most severe form of a disease called beta thalassemia to the deformation of the head of the patients. So around 7% of the world's population are actually carriers of mutations in genes that could cause hemoglobin disorders, and about 300,000 to 500,000 babies born every year are actually generating these symptoms that have to be treated. And the treatment looks as following. They are dependent of lifelong blood transfusions. Those are connected to very painful iron chelation therapies, but they also need regular bone marrow transplants in order to keep the healthy cells dominating the system. So this is a lifelong treatment that is only tackling the symptoms, but doesn't pose an actual cure to the sickness. And kind of driven by the severity of these hemoglobin disorders, scientists were trying to find a solution in order to relieve the patients and make them independent from these blood transfusions and bone marrow transplants. And in their search, they were kind of successful in the last few months. And on their search, they were inspired by nature, and to be more precise, by a bacterium called Streptococcus pyogenes, which ironically is a human pathogen that induces hemolysis, meaning that it destructs red blood cells. But this bacterium evolved a system, an immune system, that defends it from bacteriophages. And this defense system is called CRISPR-Cas. CRISPR being short for clustered, regularly interspaced short palindromic repeats. And probably from that name, you already know why we use the more crisp sounding name, CRISPR. And this system works as following. So bacterial cells are constantly under attack by small viruses called bacteriophages. And these bacteriophages attach to the bacterial cell and inject their DNA, harboring the replication machinery of the bacterium in order to replicate itself. That eventually leads to the death of the bacterial cell. But to defend itself, the genome of the bacterium contains the so-called CRISPR array, which is, you can imagine it kind of like a memory of previous viral attacks. And this memory works as following. The viral DNA gets recognized by the system and gets inserted into the CRISPR array. So basically being an exact copy of the viral thread that is now in the genome and can be used in order to defend the system, and saving it when the virus is attacking again. So now when there is another viral attack, the CRISPR array can actually be, or the information saved in the CRISPR array can be translated to a guide that is identical to the DNA that was inserted by the bacteriophage, and called the CRISPR RNA. And this CRISPR RNA is bound by Cas9, the molecular scissor from the CRISPR system. This Cas9 scissor gets cocked and triggered by a trans-activating RNA called tracer RNA. And together, the Cas9 complex is recognizing the DNA of the virus in a sequence-specific manner. And now to rewind a bit, because most of you, or some of you might have forgotten about their biology lessons and previously. So the genetic code is basically composed of 4 basic units called the bases. We have the bases A, T, G, and C, where A is binding to T and G is binding to C. And having those bases in series generates a unique fingerprint for the specific organism called the genetic code. And this genetic code is very specific for the different organisms. And now we have the CRISPR RNA carrying an exact copy of the viral DNA. So what happens now is that the region that is identical to the viral DNA is binding one of the strands, allowing Cas9 to access both of the strands, cleaving those strands and destroying the DNA, and therefore defending the bacterial cell from the viral threat. In 2012, Jungfer, Doudna and Emmanuelle Charpentier made their groundbreaking work on CRISPR-Cas, together with the contribution of my current supervisor, Martin Jinek, as Lou already told you. And what they found out was that a specific region in the CRISPR RNA called the seed sequence is essential in order to provide a sequence-specific cleavage of the target DNA. And this seed sequence is again the copy of the DNA from the virus. Moreover, what they achieved was to combine the CRISPR RNA and the tracer RNA to form the single guide RNA that can then be used as a very easily modifiable tool that is not only highly specific, but also very versatile because you can choose any seed sequence you want using molecular biology techniques. And this, in the end, is the main point for receiving the Nobel Prize in 2020. So how can you now use CRISPR-Cas to do gene editing? So you have in the first step, you have to design your CRISPR, the single guide RNA, and specifically you have to design the CRISPR, the seed region, in a way that it specifically targets the DNA that you want to cleave. Then Cas9 is bound to the target DNA. It opens the two strands. The seed regions can form complementary base pairs, and this induces the cleavage, so the cut of the DNA, leading to two loose ends. So now we have the cut DNA, but however, we still don't have any genes edited. This is where the DNA repair machinery of the bacterial cell kicks in. There are basically two mechanisms that can be used to edit genes. One is based on homology-directed repair, or short HDR, where you supply a sequence, a random sequence that you designed in a way that you want the sequence to be, together with two regions that are homologous to the sequences surrounding the gap in the DNA. The host repair machinery is now taking this homologous DNA and inserts and fills the gap that was left by Cas9 by inserting the DNA that you provided. And with that, the scientists can actually rewrite the genetic code of the organism. So scientists at Harvard University are now trying to use this method in order to resurrect extinct species. And the most prominent example of that is the woolly mammoth. And what they do is taking the DNA of frozen mammoth DNA and combining the sequences with Asian elephant DNA, creating a mixture of both that equals the DNA of the mammoth. This can be taken and reintroduced into reprogrammed skin cells that now have the function of stem cells, so they can replicate themselves and form the embryo that then grows in an artificial womb that eventually leads to the resurrection of this woolly fellow. But now back to the problem of the beginning, the genetic disease. And for that, there's another mechanism that can be used in order to, or that was used in order to tackle the hemoglobin disorders. And this is called non-homologous end joining. And in this case, you don't provide a DNA, but the host DNA repair machinery is actually trying to fix the DNA double-strand break itself. However, this mechanism is highly error-prone, meaning that it often leads to mistakes when repairing the two DNA strands, meaning that bases can be added, exchanged, or also deleted from that. And this you can imagine as a type of shut-off button that leads to a knockout of the gene. So the information encoded by the gene cannot be read properly anymore, and therefore being silenced. This was also used, or just to give you an overview, these gene knockouts have a great implication for basic research, as they give us the opportunity to analyze the function of different genes in an organism. By knocking them out, we can actually observe the effects of the gene knockout on the entire organism, and then we can conclude what the function of the gene is in this organism. But this is also the mechanism underlying the treatment of the hemoglobin disorders. In 2019, Vertex approved the first drug, CTX-001, that is based on gene editing for phase 1-2 clinical trials. And this was a huge win for recreational medicine. And this therapeutic works as following. So in the patient's cell, in the patient's bone marrow, you have the hematopoietic stem cells that generate the red and white blood cells. And these are extracted from the patients and then targeted for gene editing. And what do they do? In those hematopoietic stem cells, there's a gene called BCL11A. This encodes a repressor of fetal hemoglobin expression, meaning that it inhibits the formation of this fetal hemoglobin, short HbF. Now, the idea is to use CRISPR-Cas9 to knock this gene encoding for this repressor in order to re-achieve the production of this fetal hemoglobin that then eventually leads to the formation of normal blood cells. These are then reinserted into the patient's bone marrow, where they then boost the production of healthy blood cells that should exchange the defective blood cells. You can, going back to the example of the beginning, it's like the office mailman that was substituting the sick mailman gets now exchanged by a mailman that was retired before, but already knows the job. He knows how to drive the vehicle. He knows how to drive, where to drive, and how to supply the customer with the packages. And this is the fetal hemoglobin in this case. And extremely interesting, 2 months ago, they published the first results on this therapy, indicating that after 5 to 15 months, 50% of the fetal hemoglobin exchanged the defective hemoglobin in those patients, making all 3 patients independent of blood transfusions. And this is a great leap for recreational medicine again. Even though it's only based on preclinical or early stage clinical trials, it's still a big prospect for the future of CRISPR-Cas and its usage in medicine. So to briefly sum up what I was talking about, so I gave you an example of the importance of CRISPR-Cas in basic research, where I just briefly mentioned that we can edit genes or knock out genes and analyze their function then in the specific organism. I also told you about the de-extinction of organisms like the woolly mammoth, but also there are different other opportunities. Then we can also use CRISPR-Cas in medicine in order to treat genetic diseases that until now were not curable. But there are many more examples of the application of CRISPR-Cas. For example, tackling the challenges surrounding COVID-19, where we can use CRISPR as a kind of fast detection system in order to make all of our measurements more efficient. There's also a big influence in agriculture, where gene editing is a long used method in order to change crops in a way that they produce their fruits in a more efficient way. And this is also one of the aspects that will bring the importance of CRISPR much further. But then one thing that I unfortunately didn't have time enough to talk about is also the ethical debate. Should we even use CRISPR to do all these things? And I can assure you that behind all of those things, there's a long debate in science to talk about the aspects, the benefits, and also the negative aspects of the CRISPR-Cas usage in editing the respective genes. Unfortunately, I wasn't able to talk about it, but I'm happy to answer your question regarding that in the Q&As later on. If some of you got more excited about CRISPR now, here's a list of very interesting links that lead you to the different aspects of CRISPR that I presented to you now. With that, I thank you very much for listening, and feel free to ask your questions later on.